Yeah, so it works. Thank you very much. A very good afternoon to all of you. Basically, the kind of work we do is experimental and clinical work, and we absolutely do not do any mathematical exercises to interpret or extrapolate or justify our observations. Very minimum theoretical work that we do at our discourse. So, if you do not understand anything, just ask me. Also, I cannot have any explanation for any mathematical point of view at the moment. Okay. So, linking bacteria to cancer, it is a pretty modern and contemporary aspect of understanding bacteria and their role in short circuiting the normal homeostatic signaling of the human body. Humans and microbes have never been at war because they have been co-evolving for the millennia. It has been so recently that some of the aberrant microflora has gained entry to the human bodies and their food chain, and that is because of intense human activity and industrialization. We have been using a lot of fertilizers in our field and lot of preservatives in the feed or food, and lots of antibiotics in our gut we are putting, and that is how this may have perhaps given rise to the emergence of aberrant microbiota that may be capable of deranging the normal homeostatic signaling of the human immune system. Say for example, some of the bacteria could develop capability to make the cells resistant to apoptosis. So, as a part of homeostatic function of the human body and the bodily development, some cells must die when they achieve senescence or when they are not required. Okay. And so, when they are not able to die or when they are not put to apoptosis, then they will proliferate, and uncontrolled proliferation is what is called as cancer, or the sort of uncontrolled mitogenic activity of human cells. So, just to give you a glimpse that the microbes are not so harmful generically and traditionally to the humans, but they are their friends. I know you know better than me, maybe perhaps in the backdrop of the campaign in favor of hygiene hypothesis, we have been taking bath by the antiseptic soaps every day, Dettol and Savlon, and then antiseptic gargles and mouthwashes, and now antiseptics are everywhere, including the laundry and on the cloth and on the hands and in the car. But as a result of this, perhaps our original microbiota must have long vanished, and we may have populated an aberrant microbiota that is all playing into the game. Some of the significant functions of the microbiota include signaling related to the brain function, and this gut brain axis is now a very apparent observation that some of the bacteria in our gut, they secrete important molecules such as serotonins, and some of them they trigger through dendritic cells the cytokine production that again goes into optimization of brain function and cognition, and some of the bacterial metabolites directly also have effect into the brain signaling. So, it is a gut brain axis, for example, that is very fundamental to the functioning of human system of cognition, from digestion to cognition. And to your information, human gut is populated with 14 trillion cells at a given time, and these 14 trillion bacterial cells could weigh around 1.5 kilograms, and that is to the size of our liver. So, can you imagine the coding capacity of 1.5 kilo of bacterial cells when we gauge it in terms of chromatin, human chromatin versus the chromatin of these bacteria. It is a huge coding capacity in terms of gene gain, total gene content, total regulatory repertoire of these bacteria. But then we deal with the pathogens of the gut. We do not deal ourselves with the general normal microbiota. Among these microbiota are the pathogens which take the upper hand, and then they short circuit all the normal homeostatic pathways. They grow and outcompete the normal microbiota, or they subvert the protective effects of normal microbiota, and then they derail or derange the entire gut signaling or the immunological signaling pathways by virtue of their toxins or their virulence factors, or sheerly because of their size. For example, there is a pathogen called Leptospira. It is actually a pathogen that has a huge size, and it has an activity by which it burrows like a corkscrewing burrowing action. And when Leptospira grow in size and then when they divide uncontrolled, they block all the human blood vessels and capillaries, causing systemic hemorrhages and multi-organ failures. So they have different aspects of virulence. But then these are some of the pathogens. They stay for a very long time in the human body. We call them as chronic pathogens. So how to become a chronic pathogen? So there are some certain things to remember. EDTA is a very common chemical in our laboratories. So we try to teach to the student to remember by this word. So that means ensure adhesion. First, the pathogen must cling to the epithelium. When it is going inside the body, it must find a foothold. For us also, for example, when we are in a local train, we struggle to find a foothold and a handle to hold in a metro train or a local train or on a public transport, and then we try to sort of make ourselves stable. So pathogens also, when there is a gut motility and gut homeostasis happening, the gut is churning everything upside down, and so bacteria need to cling and adhere. So they are looking for something to adhere. So adhesion is paramount to gain a foothold for the pathogens. So there are so many molecules called adhesion molecules, which are also part of the virulence arsenal of these bacteria. And these adhesins express on their surface. And then in parallel, the human epithelial cells also have certain receptors which are fine-tuned in evolution in order to allow these adhesins to interact with the epithelial receptors. And when there is a suitable interaction actually achievable in terms of complementarity of these adhesins and the cell wall and the epithelial receptors, so bacteria try to get an adhesion-like situation. When they adhere, they also invade. So invasion is like penetrating deeper, then dig the niche, survive inside the niche in the gut or wherever, in the oral cavity or in the bloodstream or in the organs or on the skin or a scalp or anywhere. And that digging is for the carbon source. So they are all looking for a carbon source. For example, there is a pathogen called Brucella abortus, and so the name itself tells that this pathogen causes abortions. And so the destination is the placenta inside the gravid womb of a mother who is pregnant. And why the abortion should occur? Because the organism is looking for a particular sugar called erythritol, and erythritol is abundant in placenta of the human or of the animals, and that is where they are destined. And when they try to consume this sugar and they infect and multiply, there is degeneration of the placenta and abortions occur. So that is how is the modus operandi. For every pathogen there is a carbon source and preferred carbon source selected in the evolution. Then if there are any innate defenses of the body, then try to thwart them through fooling the immune system or subverting the immune responses, and then avoid clearance as long as possible, and so that they can stay for, let us say, 10 years, 20 years, 30 years, 40 years. Helicobacter pylori is the organism which I am going to talk about, which stays for the lifetime of an individual. So infections usually occur in the childhood, and then they stay for rest of the life of the individual. Two organisms are very dear to us. The first one I won't talk today. This is Mycobacterium tuberculosis, and it evolves through vertical genome minimalism. The genomes of the predecessors of these organisms are very big in size, large genomes. And then some of their very modern descendants, relatively very young in terms of evolutionary time, they have very small genomes. So there has been a vertical genomic minimalism or genome miniaturization that has been the predominant evolutionary force in mycobacteria. So they tend to downsize their genome. As they downsize, they try to adapt. They become more parasitic in the sense they depend more on the human metabolic systems for their survival. And that is how these organisms are mostly intracellular. That means they go into the cells of their host and take advantage of the host metabolic machinery directly rather than using the system of their own in most of the cases. Helicobacter, on the contrary, evolved through lateral genome acquisition to optimize their fitness. What is lateral genome acquisition? Is to acquire new genes, new genetic elements through recombinational processes. Genetic recombination is an ability of bacteria to aggregate their genome whenever they find proximity, in their proximity, some genetic material which they can import and assimilate in their genome. There are various mechanisms. We don't want to go into detail, but Helicobacter pylori is such a notorious organism that it recasts its genome every 40 years, and that is how is its acumen for acquisition of foreign DNA. And in laboratory terms, we say that if you put naked DNA onto the colony of a growing Helicobacter, it will uptake it. And that much is the recombination capacity of this organism. But amid this recombination, there is a lot of conservation also. So when new genes are imported, where are they directed? There must be some place, at that place they are stored or archived. So this organism nicely does, without disbalancing its evolutionary heritage, which are essentially in terms of housekeeping genes or the conserved genetic elements. Without compromising that content, this organism is nicely directing all these imported genes into another compartment called plasticity region cluster. And so word plasticity means this region suffers a lot of make and break, lot of evolutionary recasting in terms of evolution of new functions. So all putatively novel function encoding genes that are acquired from the environment are directed to this temporary region called plasticity region cluster without disturbing the core cluster, which is the core housekeeping function encoding compartment of the genome. This we reviewed for some time back in Nature Reviews Microbiology. You can read it, or I can send you a copy. It's a very nice review which has received around 150 citations until now. So when they are evolving through different dichotomic processes or quite opposite processes, one through aggregation of the genome and one through minimalism of the genome, and you see when you analyze them at the population level, these are hundreds of isolates of Mycobacteria and Helicobacter, Helicobacter on the top and Mycobacteria below, and just by seeing the fingerprint itself, you can make out that one has grown very clonal. One column top to the bottom is one strain, and these all are whole genome fingerprints. And so you see Mycobacterium is very highly clonal, whereas Helicobacter pylori is very diverse. These are global samples from at least 5 continents. But then Mycobacterium tuberculosis is limited to inflammation of the lungs, and that is, we call it as chronic granulomatous inflammation, where they are encapsulated by the body and then they bug the body cells, and the cell itself becomes an antigen. And the whole body cell that has been infected by the Mycobacterium tuberculosis bacilli behaves like a super antigen, and there is a huge immune response of the body, and that is how the lung is destroyed. And then there is a long, very long process of clinical morbidity of tuberculosis, and that is how is the pathology of MTB. But one thing I would like to tell you, that chronic pathogens do not aim at pathogen demise, host demise. They do not want to kill their host. And what is the purpose? because they are looking for sustained transmission opportunities. For example, if TB patient is killed the day 1 of infection, then the transmission opportunity for the pathogen is lost, because as the human body dies and decomposes, the pathogen also will die and decompose. Because of the anaerobiosis that sets in a decaying body, the gases of the rigor mortis will kill the bacterium. And that is how tuberculosis is a very long drawn disease. For 30 years or for 20 years, or at least during the course of the active disease, the patient is coughing and sneezing and infecting all other individuals, and that is how the organism is prolonging the transmission opportunities. And so that is the modus operandi, to prolong the transmission opportunities and to find new fit host all the time. And that's how they do not aim at killing their host. Okay? And that's how they are very, very chronic. And during that chronicity, it so happens that they interact with the immune system over a long time, and then somehow they end up short circuiting the normal homeostatic signaling processes in the human body, that in terms of proliferation of the cells or homeostasis of the cells and apoptosis of the cells, sometimes they click badly and then the cells start dividing in a cancerous manner. And this is because of a very long clinical course or very long course of these organisms in habitation inside their host. So longer they are staying inside their host, there is a possibility of a deranged signaling pathway that they may initiate, or a normal signaling pathway that they may short circuit. These are some of the examples of organisms that have been putatively linked to gastric cancer or to human cancer. Some of them are now very well established. Helicobacter pylori leads to gastric adenocarcinoma, chlamydia trachomatis, cervical cancer, salmonella enterica, gallbladder cancer, and propionibacterium acne, skin cancer, and E. coli, which are PKS positive, that is polyketide synthesis island. If it is present in E. coli, then they are possibly the carcinogen encoding bacilli and responsible for the colon cancer risk. Some of them are just the risk. For example, salmonella and E. coli are just the plausible linkage or risk factors, and there is not much clinical data that has accumulated. But Helicobacter pylori is very well established. It is classified as class 1 carcinogen by the WHO, and it is responsible for 39% of all cancer cases. Okay, and gastric cancer, you know how much, 500,000 cases every year of the gastric adenocarcinoma, and that is how Helicobacter has been a very classical example of cancer causing bacteria. Huh. Yeah, but correct.
A precursor to the cancer.
Yes, yes. I will give you the hierarchy of how it progresses, because there are certain host factors that they contribute to the cancer susceptibility. For example, there is a polymorphism in the human body called IL-1 beta polymorphism, and certain individuals who are actually homozygous recessive for this polymorphism, they turn out to be having very high risk of stomach cancer caused by Helicobacter. Japanese have a very high risk of Helicobacter pylori induced gastric carcinoma than the Indians, and like that. So I will tell you in greater detail as we go through the course of this. The common mechanism of bacteria to cause cancer could be due to the bacteria at their own. They initiate lot of signaling processes and then they eventually block apoptosis and induce proliferation, or could be the toxins that are encoded by the bacteria, and then these toxins create genotoxicity and thereby again the curtailment of normal signaling processes and blockage of apoptosis, or some of the products of bacteria which are actually, say for example, the nitrosourea or nitrosoamine and those kind of things, they again are highly mutagenic and then they lead to cancer. So bacteria themselves, the bacterial encoded toxins or the metabolic products of bacteria, they are broadly responsible for triggering the cancerous growth. I can give you an example here. Do not go into the details of this slide, it's a very loaded slide. But you see the references here are path breaking studies published in Science in 2006. First linkage of E. coli to the double-stranded DNA breaks in human cells, HeLa cells. And you see, this is something we call it as COMET assay. The COMET assay is a gold standard or a signature test for identifying the double-stranded breaks in the human DNA, or for any DNA for that matter. And here you see the control doesn't have any comets here, but then but then you see that adipocyte, which is a known carcinogen, and the back PKS, that's a back vector that has been cloned with the PKS island of E. coli, has actually induced DNA damage here. You see the normal cells, and you see the cells with deranged morphogenesis control, and here the same thing that the Salmonella does through a CDT, cytolethal distending toxin, and then again it puts into anchorage independent growth. When the cells in vitro, they grow through anchorage, they stick to the plate inside the laboratory. We have a cell culture plate, and on that cell culture plate, cells, they divide and then they multiply in an anchorage dependent manner, and that is how is the phenotype of normal cells in culture. But when they lose anchorage, then they multiply in an anchorage independent manner. That is something, an abnormal growth pattern, and that is what is induced by the Salmonella and E. coli in all these assays. They cause the double-stranded DNA break because of the polycateide toxins that they include, and this toxin is highly toxic to the integrity of the DNA. Okay? And when the DNA is broken, then there are certain markers that we have. Say, for example, the histone deacetylation related markers that can be scored in these cases in order to know whether the DNA has indeed been damaged, apart from the physical tests such as the COMET assay. The same thing here involving all other different organisms, different strains of E. coli, and different of Klebsiella pneumonia, and another strain of E. coli, and you see the arrangement of the PKS encoding genes here in the genomes of these organisms. And very characteristic here is you see the normal control, you see the behavior of the cells here, normal staining of DNA, and the cell cycle clearly separating G1, G2 phase, whereas in all other cases there is a arrest in G2 phase and a prolonged G1 phase. Coming back to Helicobacter again, after giving these examples, of course these are some of the very strong linkages, very nice recent papers giving mechanistic insights to what happens and how cancer occurs by bacterial signaling. We call it as a celebrity bug because I have not seen a single pathogen where you have a journal in the name of that pathogen. We have a journal called Helicobacter, and very respectable journal having impact factor 4. And a Nobel Prize was given for the discovery of this organism 100 years after the cause and effect relationship of Mycobacterium tuberculosis to tuberculosis by Robert Koch. In 1905, the Nobel Prize was given for a cause and effect relationship. Exactly after 100 years in 2005, Nobel Prize was given for a cause and effect relationship to Barry Marshall for identifying the cause of gastritis or gastric ulcers or duodenal ulcers or gastric adenocarcinoma. And that is how is the Nobel Prize for this. And there is a very vibrant scientific society, the European Helicobacter and Microbiota Study Group, again established for this organism. Then there are certain things like there is an established link with gastric cancer. This has been now established. Helicobacter is no more a risk factor, it is a causative agent of gastric carcinoma. Cause and effect enigmas in developing countries, of course. In India, for example, we have up to 98% colonization. Anyone can test positive any day. If you go for an endoscopy and take a probe or take a swab from the endoscope and culture it, you will get Helicobacter positivity. But not all Indians suffer from the overt outcome of Helicobacter colonization. The rate of clinical ailment is very, very low, maybe 10%. And our gastric carcinoma incidence in India is 3 per 100,000, whereas in Japan it is 75 per 100,000. Can you imagine? So developing countries actually have this enigmatic situation where high rate of infection doesn't translate into high rates of adverse outcome of the infection. Then you have global consumer frenzy in terms of taking antacids. So it has given huge business to the companies which manufacture antacids, chewable antacids and all other sorts of things that are available on over the counter. It creates a huge market for this kind of things. Here is the course of Helicobacter infection. There is a lot of genetics that contributes. Up to 50% I will say that there is a genetic contribution. Host genetic factors, virulence factors, host immune responses and the bone marrow derived cells. All these contribute, and then normal gastric mucosa, whenever infection happens, there is an acute infection which is rare. 100% people, they have chronic infection, and out of this only 20% get duodenal ulcer, and only 10% of those who get the ulcers, they progress to gastric ulcer, and then eventually the gastric adenocarcinoma is again very, very rare here. Up to 2% of that infected compartment which are chronically infected and that have developed gastric ulcer, then they progress to gastric cancer over a period of time. So 90% of the people should be asymptomatic H. pylori carriers for whole of their life. There is a lot of greener side to this organism. There are people who are for and against eradication. There are some school of thought, they say why eradicate an organism which is harmless in 90% of the cases? Why eat antibiotics unnecessarily of 10,000 rupees sometimes and over a period of 1 week? And some people develop very serious complications of this eradication therapy. Some report high blood pressure and nausea and a lot of complications. So why go for eradication when it is anyway harmless? In 90% cases, it doesn't matter. H. pylori is co-evolved with humans and is a marker of human biology. Eradication brings new problems such as gastroesophageal reflux disease. So when you eradicate it, then you develop something called as reflux disease, because Helicobacter is a natural dump for your excess acidity, because it has a mechanism by which it actually breaks your saliva into bicarbonate and ammonia by using its urease mechanism. And so that is how the excess acidity of the stomach is dumped by Helicobacter. But then when it is eradicated, in eradicated cases, severe gastroesophageal reflux disease has developed. That is again a serious risk factor for esophageal cancer. Okay? So colonization does not lead to a severe outcome in some countries. That we have already discussed. Recolonization is imminent after eradication. So if you eradicate it, you cannot prevent recolonization, because everybody else in the family is also infected or colonized asymptomatically. So usually spouses, they freely contribute to reinfection, and another contributory factor is the drinking water, because all of us do not drink packaged drinking water. And in developing countries, quite often the sewage is mixing with the drinking water because the lines are laid down parallel, and I don't know how, but then recolonization after eradication is imminent because it keeps coming. So when all these things happen, why eradicate first of all? That is the debate. But in Japan and all other countries, eradication is actually the policy of the government. They will immediately eradicate and put you on the antibiotics for some time. So here is the key of persistence. Don't go into the detail again, there is a hugely loaded slide here, but then that is the reality of bacterial infection as to what happens. This is the blood vessel, and this is the mucous membrane, and this is the submucosal space where all sorts of this drama happens with respect to recruitment of the cells of inflammation under the chemotactic influence of some of the virulence factors that Helicobacter is secreting. Very important here is that two counterbalancing virulence factors, CAG and VAC, just remember them. They are the cardinal virulence factors whose activities are opposite to each other. One is very highly pro-inflammatory and another one is pro-apoptotic. So pro-inflammatory will lead to proliferation of the immune cells and more and more inflammation, and the another one will lead to more and more apoptosis, and so there is a sort of balance achieved. At some point of time there is homeostasis or a net balance where the organism will persist without being cleared by the host immune system. And this CAG-A and VAC-A, so first to begin with the CAG-A protein here, it's a toxin. The name is cytotoxin associated gene A. That is the full form of CAG-A. And you see it has been nicely captured here when Helicobacter is delivering the CAG-A into the human cells. So the CAG-A must be injected into the epithelial cells. If it is not injected inside, it is not functional, because it has to be phosphorylated when it is injected inside the human cell. And for injection, there is a special apparatus. Can you see here a complicated machine and an injector here, a kind of a what is that, maze-like structure here. It is made up of so many proteins. 27 different proteins come together to make that barrel-like syringe. When you go to the doctor, there is a syringe and there is a needle and there is a piston, and inside there is a molecule that is being injected. And the same way Helicobacter is using the syringe and a needle and a molecule inside to inject into the epithelial cells. And this has been stained differentially to show how Helicobacter is injecting CAG-A inside. When it injects the CAG-A, there are epithelial tight junctions, the cellular integrity and cell polarity is lost, and then motility, invasiveness and all these kind of things. Eventually, over the years, the carcinogenesis develops. And this is how the Helicobacter CAG-A is going inside, and then it is phosphorylated. Phosphorylation is again dependent on the type of CAG-A signature motifs. So in Japanese, you see arrow here. This motif is very predominant, which we call it as the APIA D type of phosphorylation motif, which are predominant in Japanese Helicobacter pylori, and all other Western type, they are not very dangerous. They not always lead to gastric carcinoma, but then this one will for sure lead to very high rates of phosphorylation of CAG-A, and thereby CAG-A is very active in participating into the carcinogenic signaling. Another thing is that because of this, the CAG-A will dissolve the epithelial junctions and give lot of signaling, pro-inflammatory signaling, and then as a result, these things will happen: deregulation in mitogenic signaling, apoptosis, cell scattering, and there is something very typical called hummingbird phenotype. So CAG-A is very specifically leading to hummingbird phenotype, and this hummingbird phenotype is an early precursor and a marker of cancer growth. These cells are now getting into endless multiplication cascade. When somebody tried to make a transgenic mouse expressing CAG-A protein in mice under a constitutive promoter, what is this constitutive promoter is that when you clone a gene under this promoter and put into the germline, this gene will express in all organs of the body. And so when CAG-A gene was cloned constitutively under a very nice promoter in mice, you see that in all tissues mice developed tumors, especially the spleen, the mesenteric lymph nodes, the liver, lung, and all other cells, all other tissues of the gut and the lung and the spleen, and there was also leukemia. So a series of cancers were caused by this protein. That was the first evidence, direct evidence in a mammalian species that CAG-A is actually a oncoprotein or a microbial oncoprotein, which is very dangerous in itself because it can cause a sort of cancer irrelevant to any body tissue. Of course, for Helicobacter, the niche is traditionally the stomach, and that is how it links to the stomach cancer. And if this organism could have traveled elsewhere and developed its niche, then it is anyway capable of causing cancer there also. Another virulence factor is VAC-A, vacuolating cytotoxin A, and these are some of the things that it causes: pro-inflammatory signaling, increased permeability of the plasma membrane, and alterations in mitochondrial membrane, and you see it is having a different phenotype. Previously we have seen a hummingbird phenotype, here it is a vacuolation phenotype. So it will create vacuolations in the cells. So cells will bubble up like they will have lots of bubble inside, and that is how is this phenotype. But then there is a dichotomy in understanding when this is so widespread organism that everybody is having it in their stomach. Has it been always associated with humans? From where it has come? That's the question. When we say something has co-evolved with humans, so something that is co-evolving should not be so dangerous. It should have been adapted to the body system of the human beings, as like other organisms have adapted, like our normal microbiota has adapted to ourselves. And so when these organisms are co-evolving, I mean, why should they be so pathogenic? So there is a hypothesis which needs multiple evidences at different levels. The hypothesis here is that these virulence factors were not always in this pathogen. This has been a benign organism insofar until human beings started living together with animals, and that was in the Neolithic period, when the domestication of animals and domestication of crop plants would have happened. And this scenario explains that microorganisms in early cultivated plants, domesticated animals, pets or rodent pests must have contributed these virulence factors to Helicobacter. So here is a non-pathogenic colonizer, and when the agriculture arrived in a continent-specific manner, the continent-specific populations of Helicobacter with this recombinational genotype emerged in different continents, and that is how there has been a global rise of acid peptic diseases and gastritis and gastric ulcers and duodenal ulcers and whatnot. And that is the hypothesis so far, that these Kage and Vaccae have never been with Helicobacter since the beginning. They have been acquired in the evolution of the organism, possibly because of its proximity with all other organisms. And we have some patchy evidence that there is an organism in plant microbiology very famous called Agrobacterium tumefaciens. And as I have shown you, the barrel-like structure of Kage here, the similar structure is found conserved in Agrobacterium tumefaciens, also called as Vir barrel, the virulence related barrel of the Agrobacterium tumefaciens. I am not showing you that data, but that hints that sometime this was acquired during evolution by Helicobacter, and that has given it a power to become a carcinogen. When we say it has been co-evolved, means it has same geographical compartmentalization as the human populations have. And you see the evolution from West Africa, and then nice, very large Indo-European branching, and then this is African branching, and this is East Asian branching. So it has a very nice geographical compartmentalization that is indicative of how this organism has co-evolved with its human host. And that's how it has a very tight relationship with the human host. Now, of course, there is evidence in the form of genomes that are being sequenced. Of course, up to 3,000 genomes of Helicobacter are at our disposal now to pool, and then as these new genomes are being sequenced, you see the relationship. The core gene pool is stabilizing, whereas the accessory gene pool is always rising. And as I told you, that those organisms which recombine and uptake genetic elements in a lateral fashion, they will always have novel genes to their credit. Whenever a new genome is sequenced, new genes are found. And by probability, each new genome should contribute 40 new genes. And that is how every single genome is welcome in those species whose pan genome is open. So Helicobacter is one of those species where the pan genome is open. And so every new genome sequence will add new genes. So what these new genes are doing, and how to put hold on these new genes. It is always difficult to extract new genes, so you need new tools. So we have developed a CL authenticator, an improved genomic assembly algorithm for bacteria, and you see that the troubled regions are very nicely resolved by our tool. This has been communicated, and a second round of revision is going on. But in summary, I should tell you that with the help of this software, it is possible to identify new genes quickly, rather than struggling with all other small tools. So with that hypothesis that every new genome of Helicobacter will give you the new genes, we have sequenced several of the genomes from Helicobacter pylori in Malaysia. Why should we sequence genomes from Malaysia? Because Malaysia is a country where we have huge diversity. 3 distinct human populations are there. There are ethnic Chinese and ethnic Indians, and there are Malay. So Malay are a hybrid population, Indians are pure population, and the Chinese are pure population. And when they are in a melting pot of a common culture, in a common kitchen, and in common customs and traditions, how these organisms behave, which are so far geographically very highly compartmentalized. So we went to Malaysia, we sequenced the genomes from there, we published in Nucleic Acids Research this year in January. But the point is not that. Point is to tell you that each new genome that was sequenced with respect to the core gene pool, there were 8, 14, 18, 11, 13, 5, 13, these many new genes were found per genome. Of course, they were not complete genomes, they were draft genomes, and that is how the number of genes were less, because we didn't have confidence in identifying more number of genes. So whatever genes were confidently identified, they were all novel genes, hitherto unreported and unrecorded. But that is about an independent genome. What about the bacteria that are inside the stomach of an individual, how they evolve? When they are evolving in a community, of course, but while inside in the stomach of an individual, how Helicobacter evolves? As I told you that it recasts its genome every 40 years, and then it comes up with novel combinations of genes. We looked at this individual. After this story, I'll end my talk in 5 more minutes. This is an African individual living in France. So don't look for him on the Facebook if you can, but we are not supposed to give his name and his face. Okay. And this person came to our collaborators in 1994 and in 2003, 2 occasions with the same disease. And so on first occasion, bacteria were recovered and ulcer was identified, the drugs were given, the person recovered and went back. And so those organisms were frozen, okay, and they were cultured for a limited time of point, and then he was sent away. Exactly 10 years after, the same person came with the same complications, and then again the organisms were recovered. And they were the same organisms, basically, same complaints, same individual, and same situation. And the drugs were given and again went back nicely because it was eradicated, recovered, and went back so far healthy. But then what was the lesson? The lesson was we had organisms at inclusion before 10 years and after 10 years, and we had an opportunity to look at chronological evolution over a decade in these organisms which are evolving in a single patient, in a single stomach, evolution of a single bacterial strain. We could publish a lot of data from this individual. These are some of the papers that you can read. But then I will also show you how the evolution has happened. So the first thing the reviewers actually confronted us was, what is the guarantee that these were the same isolates that you recovered after 10 years and they were not the entirely different stock reinfection? It's possible a person in his or her life may get reinfected again and again by many different strains. But the beauty with Helicobacter is that you will have many strains coming to your system, but as I told you that Helicobacter adhesins are looking for complementary receptors on the human epithelial cells. When there is a match of adhesins with the human receptors, then only they will get a foothold, otherwise they will move away. And that is how there was for sure that these were the same organisms that we recovered. But then they wanted us to prove that these are the same organisms, so we did a lot of fingerprinting with ERIC PCR, REP PCR, RAPD PCR, all these are known tools in molecular biology. That with this it was found that they were the same isolates. Then we did genome sequencing of these isolates, 1 at inclusion, 2 afterwards. They all were same. Then we did something called multi-locus sequence typing, and they all clustered here together. And that is how we looked at the same stock after 10 years. This was the evidence. But then the question is, when this fellow was given treatment, where did this organism was hiding for 10 years without any symptoms? So the answer is that it is possible that one of the individual in the family is asymptomatically carrying this organism, perhaps the spouse. And that is how, since the spouse was healthy, our doctors did not have opportunity to take her samples, because you cannot touch a healthy individual just out of curiosity. So curiosity doesn't help there, because these things are governed by ethics. And you see that all these organisms were similar. 908 10 years before, and these two after 10 years, and they all were similar, demonstrating the propensity of this organism to survive for as long as 10 years and again come back. And that is how Helicobacter is evolving. There is a lot of wet lab data which I am skipping. I must tell you that the conclusions are very important, and let me go to the conclusions straight away and take your questions. But before that, I will tell you something about these publications. So this is the comparative genomic analysis of Helicobacter from Malaysia that has identified 3 distinct lineages suggestive of differential evolution. Even if culturally they are intermingling and getting homogenized, the Malay harbor a distinct group of Helicobacter, then the South Indians, then the Chinese. And that is the message. And you see that this Barry J. Marshall is none other than the Nobel laureate of 2005, with whom we have published this paper. Narendra Kumar has done a lot of work here. He is the PhD student who has now submitted his thesis and waiting for his viva, because the external examiner is not able to confirm the dates. Okay? And this this author has already secured a postdoctoral position in University of Cambridge. And so that is how is the system that works, that he is waiting desperately for his viva to be done so that he can fly. Okay. This is another paper in Scientific Reports which has come in October this year out of the work that I have shown you so far. These are the summary pointers. Genomic analysis offers a robust tool to glean new genes, new functions, new lifestyles shaping up in different hosts over a period of time. Bacterial genes, proteins could potentially interact with the host system. Such potential interactions could mean novel adaptation functions, the paradigm of 986 and Minkel regulated survival mechanism, which I just skipped because of the short of time. I would like to just acknowledge quickly some people here whose work I have shown is Narendra Kumar, of course, who is here, and Savita Devi, another PhD student, Sohail, another PhD student here, and this is our extended group of the European collaborators, our students, our collaborators, and me here just behind Savita. Programs and support, Indian government, of course, DBT, CSIR for the grants, German Research Foundation for the graduate college, the program they call it as graduate college, GRK 1673, University of Malaya for high impact research grant, where that NAR paper has come, and the Marie Curie IRSS network EU FP7 program on mathematics in health and disease, for which I am here. Thank you so much for inviting, and I will be very happy to answer your questions.
Quick question, like clarification, is that like you said that in developing country and developed country, like for example Japan, you see that different strains like colonization of this Helicobacter. I mean, I'm trying to understand like what are the like population level process, like for example socioeconomic status or food habit or kind of what actually kind of leads to this disparity of this colonization, like any kind of idea?
No, if you say that why specific strains are destined to specific individuals, but there is a 2-tier selection, as I told you. The adhesins on the capsule of bacteria and the surface receptors of the human epithelial cells should match each other, just like plug and pin. Okay? As long as that match is found, the strain is destined to that host only. That's the one thing, because they are tailor-made for each other. And that is how, even if an Indian is staying in Japan, the Japanese strain is not going to colonize an Indian patient, and like that. And other than that, the socio-economic status, as you said, will determine how often an individual actually has a chance to get infected. Okay? And so when you are in developing country, because of poor hygiene or sanitation, or let us say the lack of adequately sterilized water or whatever, will actually colonize. Because when it depends how homogeneous is the human population. So if we say that we all are South Indians, so there should be an HP South India, which will have an equal probability to infect every one of us, irrespective of our name, because then it will go by our genetic descent. It will look for whether the receptor homology is there to the adhesins or not, straight away. And so if we all suffer of a similar socio-economic status, suffer or enjoy or enrich that thing, then Helicobacter will have an equal background for all of us, a common background rather. But in Japanese case scenario, the virulence is very, very high. As I told you that there is an Apia motif D, which is very much correlating with the phosphorylation of Kage. Kage is not active alone, it needs to be phosphorylated. So either you are deficient in Kage, so your strain do not have Kage, so you will not suffer for cancer for sure. How many, even if 100 years Helicobacter colonizes, no cancer. Kage is not there. Kage is there, Kage is not able to be injected out. So the barrel is deficient, the syringe is not assembling. Even if it is injected to the epithelial cells, it is not getting phosphorylated. Okay, so these are three regulatory checkpoints. If it is phosphorylated, the phosphorylation is not very efficient. In Japanese case, all these are true. All of their strains have Kage, all their strains are able to inject Kage, and all their Kage is able to nicely phosphorylate to become a very active oncoprotein. And that is how the Apia type D motif is the signature of the Japanese Helicobacter, and hence the outcome of gastric cancer. You see 75 per 100,000, that is the incidence. Yes, please.
Since there is a lateral uptake of genetic material, could that be used against the microbe? Could that be used to sabotage some of the virulent factors in the microbe?
They have a system called restriction modification system. And Helicobacter pylori is notoriously known to encode as many as 34 different restriction modification systems. So it won't let any deleterious characters take upper hand in its genome. And that's how I said that all whatever is imported will be actually directed to a different compartment of the genome called plasticity region cluster, without actually compromising the core gene functions. And from that plasticity region cluster, they will be co-opted for different functions. If that component is functional for some reason, then it will co-opt for compensation of Kage function or Vake function, in case Kage and Vake are evolving or mutated or something. Thank you so much again for your patience and trying to understand my data.
So let's break for lunch now, and it's now 1:25. Shall we meet at 2:15? Let's meet at 2:15. Should be enough time for lunch. We just move the whole program up by 15.